So we have, everybody agrees that we have 5 taste modalities. We can detect sweet, salty, salt, sodium chloride. We detect glutamate, which is it's called umami. This is why, so it's hard to describe. It's the taste of deliciousness. It's a savory taste. Meat, if you're not a vegetarian, meat is rich in glutamate. This is why people put monosodium glutamate in food if they want to enhance flavor by increasing the activation of this receptor. So, sweet, salty, umami, bitter, and sour. We detect bitter chemicals and we detect acid as sour. Those are the 5 universally recognized taste modalities. There's a cultural element to this. Some say that capsaicin, the ability to detect capsaicin, is a 6th modality, the taste of spicy hot food. So in some cultures, that's an incredible part of taste. I've told you about capsaicin receptors, and I put it into the pain category. But that's Western hegemony. Other cultures, African, South Asian, East Asian cultures, do put the taste of spice mediated by capsaicin receptors as a 6th modality of taste. They recognize it as pain, it's part of the pain pathway, but it's also part of the taste pathway. We taste capsaicin. Okay? So some cultural variance in what we taste, or what we call taste. You all know that the taste comes from the taste buds in your tongue. So this is a cartoon of a taste bud. Here's the surface of the tongue. Here is the taste bud. Here are the receptor cells in the taste bud connected to afferent nerves. So the taste, I'm going to tell you about how the taste activates these cells, releases neurotransmitter, activates an afferent nerve. Is taste long or short? Receptor cells. These are short because the receptor cell itself does not have the axon. It's the afferent neuron that has the axon. The cells release neurotransmitter. So there are different types of taste buds that I don't want you to memorize. So we have taste buds at the tip of the tongue that have a name. We have taste buds at the back of the tongue that have a name. We actually have taste buds in the back of the throat that have a name. I don't want you to know the names. I do want you to know that there are different locations of the taste buds. Tip, back of the tongue, throat. The ones, and here's an interesting phenomenon. I don't think I would test you on this, but there are 3 cranial nerves that carry taste. So it's interesting. We only have 1 that carries smell. We have 3 that carry taste. I can't explain the evolutionary heritage of that. But there are 3 cranial nerves that carry taste. One is the vagus nerve, carries taste information from the throat. The other is the facial nerve, carries taste information from the tip of the tongue. And the glossopharyngeal carries taste information from the back of the tongue. I don't know why. But you should know that there are 3 cranial nerves that carry taste. You may have heard that the tip of the tongue transduces sweet and the back of the tongue transduces bitter, and that's wrong. The tip of the tongue is better for detecting sweet, but it also detects bitter. The back of the tongue is better for detecting bitter, but it also detects sweet. The throat is best for detecting salty, but it detects all of them. So, in the taste bud, we have these different types of receptor cells that are transducing these different chemical stimuli. I wish I could tell you that they were all the same, like they are in the olfactory system, because that would be nice, right? It would be simple. The olfactory system at this level is simple. Odorants, GPCR, G-olf, depolarization, opening a cyclic AMP dependent protein, cation channel, etc. That's simple, straightforward. They're all the same. In the taste system, they're not all the same. Some of them are GPCRs. Sweet, bitter, and umami activate GPCRs that are coupled to GQ. And there's the GQ story, right? You've seen this story before. GQ, calcium neurotransmitter release. This one's easy, right? Receptor, G protein story, IP3 dependent calcium release, calcium neurotransmitter release. The next easiest one is salty. If we put salt on the tongue, the sodium goes through a constitutively active sodium channel. This is not voltage gated. This is just a leak sodium channel. More sodium influx. What does that do? Depolarizes. What does that do? Opens voltage gated calcium channels, calcium influx, neurotransmitter release. So here the calcium is from these stores, and here the calcium is through voltage-gated calcium channels because the stimulus causes a depolarization, not a G protein. So I do want you to know that sweet, bitter, and umami activate a GQ-coupled GPCR, and it uses the GQ story. I do want you to know that salt goes through a leak sodium channel, depolarizes, voltage-gated calcium channels, neurotransmitter release. So we've got 1, 2, 3, 4. We've covered 4 of the 5. What I want you to know, so this is more complicated than it needs to be, but what I want you to know is right here. And this uses some of your chemistry. The acids we detect are in combination with their, conjugate base. You've learned about acids and conjugate bases in chemistry. When the base is protonated, it's relatively amphipathic. It can diffuse through the membrane. Diffusion of this protonated base, and then it dissociates. The acid, the proton, now inside the cell. So this is outside the cell, this is inside the cell. The acid can go through the cell, not as a proton, but as a protonated base. And the proton blocks a potassium channel. Blocking of potassium channel, decreased potassium efflux, depolarization due to decreased potassium efflux, voltage-gated calcium channels, calcium release, calcium influx, neurotransmitter release. So acid gets in and blocks a potassium channel. In addition, acid as a proton can go through proton-selective channels. And this is depolarization as well. So everything is here. Protons going through, depolarization, increasing depolarization, increasing neurotransmitter release, increasing action potentials, Not of the cell, but of the neurite, of the neuron that it's coupled to. In addition to protons going through the channel, protons can get into the cell and block a potassium channel, which also causes depolarization. I think you can just sort of cross out. The rest of this is, it's the same story, it's just more than you need. I should just get rid of this. So you get rid of it on your, did I give you this slide as a handout? Yeah, just focus your attention on understanding that image. You do not have to focus on understanding this part of the image. The calcium does stuff, but the calcium is what triggers the neurotransmitter release from the cells that are expressing bitter, sweet, or umami receptors. So we have receptors. So sodium channel, this is detecting salt, sodium going in. We have G protein coupled receptors triggering calcium release. So that's bitter, sweet, and umami. That receptor, second messenger, this is putting it in a black box, but you know what's going on here. The G protein story, the IP3 story, the calcium release story, calcium causing neurotransmitter release. This slide is illustrating that whether we cause a calcium release from the intracellular stores or a depolarization dependent calcium influx through the voltage-gated calcium channels, both of those cause neurotransmitter release. The neurotransmitters are still being figured out, actually. We don't know all the neurotransmitters at this synapse, but they include serotonin, norepinephrine, and ATP. I will not test you on that. There are excitatory neurotransmitter releases that trigger depolarization of the nerve, depolarization, threshold, action potential propagation. To the thalamus and to the taste cortex. To the thalamus, I'm going to go back. Well, I don't think I have it. I have to go to the other. The taste cortex is in the other power plant. Oh, no, is it on this one? Right there, sorry. The taste cortex is in the inferior medial frontal lobe. It's a part of the lobe called the insula. I will not test you on that. I do want you to know that the taste primary taste cortex, and the primary olfactory cortex are distinct, but they're nearby. Because flavor is a combination of both taste and smell. So I do want you to know the different mechanisms, and I do want you to know that the receptors are short, causing cytoplasmic calcium to increase, causing an excitatory neurotransmitter release, excitatory receptors, depolarization, action potentials. So now, in the past, when everybody was in the room and we were, well, in the past, I had a whole series of clickers. And but this year I've been mostly assigning the clickers as homework. Let me poll the room, and those of you online as well, because you can do this too. Would you like to spend some time going through the clickers, or would you rather do it as homework? We've been in this room now for almost 2 hours. That's a long time. That's a lot of information. We can do it. We can reinforce some things with clickers here, or you can reinforce it at home with your friends or on your own. I don't have a preference. I really don't have a preference. If you want to say, gosh, 2 hours is enough, I'm ready to go home and start to integrate this after I get a nap or whatever, If you would like to go through this here with me now, I'm willing to do that. So, I know this is an awkward question for a professor to ask students, how do you want to do the rest of the class? So in fact, what I'm going to do is I'm going to go get a drink of water. You're going to reach consensus, and then someone's going to tell me what the consensus of the room is, okay? So you have about 2 minutes between for me to get to the water fountain and to get back to reach consensus. And if you don't